So these two papers for today are really about, I chose them because they're sort of a case studies of ribosomal RNA sequencing, and the latest, they literally came out last week. So this is the latest things that people are doing with ribosomal RNA sequencing. And it's also interesting because it's kind of a new arena for using ribosomal RNA sequencing. And this is basically taking all the approaches that we've talked about so far and applying them to try and study the ecology of microbes in human-built environments, like buildings and cars and planes and warehouses and food production facilities and cruise ships and so on. And people have been studying pathogens in these places for a long time, but generally the ecology of these communities has just been ignored. And I think that that's mostly because microbial ecologists have wanted to study natural ecosystems and not really cared about human built. They're new, they're not really natural places, and they've been sort of pushed aside. But then both because there's money available to do this, so the Sloan Foundation came in and said, we want to start a program in this area, and because people are realizing that the microbes that are in buildings have a big effect on the human microbiome and on health of the inhabitants, that there's been a bigger move to study the sort of everything about the microbes in buildings. And what these two papers are really about is even going the next step, which is to try and get people to think about what we're doing in buildings, either the design of the buildings or the movement of people in the buildings, or like in this hospital study, what we do in certain parts of a hospital and how that affects the microbial ecology and how that in turn might affect the health of the inhabitants in these buildings. And a lot of this right now, before, leading up to these studies or even after these studies, is still, the health connection is tenuous at best. But the idea that comes from human microbiome studies is that the microbial community in and on people can affect our health. It's pretty clear that the microbial community in and around our buildings can affect the microbial community in and on us, and therefore the idea is that somehow this might affect our health. And then there are other people who are studying it that, like this group that wrote the first paper. They are interested in human health, but they're also interested in other issues like the decay of a building, for example, and how sustainable building design is based upon microbes degrading the drywall, or if you're doing a facility for food production, that's probably going to require a different microbial ecology concept than a facility for pets or for animals. So this first study is from this group that has a project funded by this Sloan Foundation program, and their basic goal is to start to study the links between architecture and sort of big picture building design parameters and the microbial ecology of the community that is in those buildings. So what they're really interested in is things like airflow dynamics and size of a room, or the traffic in a room, or the really big picture findings. And the second paper, this paper on the hospitals that we'll talk about, is much more interested in very small scale details like what's on the doctor's gloves, or what's on particular parts of the facility in the NICU. And so these are sort of contrasting scale of the what people are trying to look at in terms of the building, the microbial ecology of buildings. So for this paper on the architecture influence on microbiology. They chose this building. I don't know if anybody's been there, if people have been to University of Oregon, this Lillis Hall. I think, they talk about some of the features of this building. I don't know if people have any questions about that or any comments about why they picked this facility. Remember, when people write papers, it's frequently you do something and then you make up a justification afterwards, just to let you know. But why did they, what are the features of this building that they liked?
It's sort of a contrast immediately because it's long and narrow, so you have a chance for pervasive natural light or air ventilation.
Yeah, and you have gradients. That's right.
Yeah, there's a gradient because then they also have a central shaft, mechanical shaft.
Right. So that's one feature. It has multiple floors. There's another feature that was really important to their study, which was that they can control the airflow pretty well throughout the building. So I think they're going to continue to study this building, and then they're going to shut off, airflow to some rooms and see what happens. And they're going to, change it from outdoor air coming into one area versus recirculating the indoor air coming into an area. So my impression is they're going to do a lot of studies of this one building and start to change some of the parameters.
Was the fact that it was relatively new also part of it, or not?
Yeah, I mean, they seem, I've never the people who did this, many of them are friends of mine, but I've never actually talked to them in detail about this particular study. And the senior author, Jessica Green, is going to be here tomorrow, actually. But I'm not sure if the age mattered as much. They hinted that it mattered, that it was part of their choice, but then they say later that, because it's 10 years old, the dust that is accumulating in the different parts could span over a long period of time. So I think to some of the people, 10 years was too old. Like they wanted to get an actually brand new building and start to study it as it gets colonized, because then everything would be recently generated samples. And other people, if you compare like on Davis, the same thing as at Oregon, most of the buildings are 50 to 80 years old. So a 10-year-old building is like shiny, sparkling new. So I think being new helped them because it had lots of features like the air control system. I think it's like a LEED Platinum certified facility, and it's, energy minimal use, and it has all these, again, features like control the shading in different parts and control the airflow and control the window openings, and makes it useful. But I think it probably would have been better to start with an unoccupied building, study it before it's occupied and do some manipulations before it gets occupied, and then let it get occupied and compare and contrast, which they have also done with other facilities.
But then it wouldn't have a chance to get a critical mass of microorganisms relatively new. Or not, it wouldn't have as many people contacting.
It might not be in equilibrium.
Yeah, right.
So they're doing both. So maybe this is one way of doing a building that's got that critical mass. And
since the 80s, more attention has been paid to vapor migration and air turnovers in buildings, which goes along with the LEED stuff.
Yeah.
In an ancient building, you're going to have drafts and dampness and that kind of thing, which is not good for some microorganisms, right?
Yeah, so, a lot of this is, there's anecdotal information or occasionally information about, say, one organism like black mold, that people have an idea about how dampness or certain airflow dynamics affect that one type of, or Legionella, the causative agent of Legionnaire's disease. They know about how it's growing in water coolers and in water evaporation systems, but those buildings, nobody's ever studied anything else in most of those buildings. So this is really about the whole ecosystem. And I think they could have done this they could have done it in any building. I think they like the controllability of newer buildings.
It's interesting that the Sloan Foundation couldn't afford afford to fund them for something besides a shop vac.
Well, so they tried a lot of different things, actually.
Oh, they did?
Yeah.
Because they have dust collection systems now with HEPA filters and all that. And a shop vac's blowing about 70% of what it picks up back into the environment.
Yeah, we should talk to Holly. We've been looking into
Oh,
exactly how to.
Oh, no, really?
Yeah.
It fit it with a filter to try
Oh, better filter than it comes with,
Yeah.
which is a piece of
Yeah.
foam stuff that doesn't catch.
So they've done this group. So yeah, I mean, you can say that about the Sloan Foundation. A lot of other people complained that this group got $5 million and everybody else who got grants got $1 million. So I don't think they could have asked for anything more in their funding. But they've done studies where they've used what are called liquid impingers, where they have, it's like a bong, basically. They're sucking the air through the water, and then they collect the water, and that's where the microbes have been sampled. And they've put some of these out in like a building. But they have to run for like a day and a half to get enough material, and then they worry that the stuff in the water is starting to degrade. And so then they tried lots of small vacuum run filtration systems scattered throughout some of these buildings, and people were moving them, and they had to cordon them off, and then they were really loud and people complained. And so they've tried a lot of different methods there's one of the news stories about it has Steve Kemble, the first author, showing him with the shop vac, like walking around the building, which is really, I don't know if I have a picture of that.
Is that the janitor or the scientist?
Yeah, well, I'll have to call him that from now on. So, I think the key thing for their study is connecting the microbiology data to building, and in particular architectural, related features about the buildings. So they, and it's not totally new, they're working with an architectural design group at University of Oregon that has been studying the buildings at University of Oregon and in other places, and has their own classification system for classifying rooms and parts of buildings into different sort of types of entities. And they go through this incredibly complicated classification system that they have, which has like 4 different layers. You can classify space type. You can classify the building support, entity and other building parameters. You can classify the form and organization of the building and many other things. And this, to them, is sort of what microbial ecologists call metadata. So you're going to end up with a lot of data about microbes from different samples. And like we talked about when people are doing the principal coordinate analysis or any other type of comparative analysis of the microbes in your samples, the main goal of most people doing microbial ecology right now is to then try and relate something you measure in that microbial community to variation in some other parameter, whether that's Crohn's disease or healthy or different ages of people, or in this case, different building parameters. And this is a massive study compared to what has been done previously. I think it may have been on the other slide. They did 155 or something rooms. Yeah, so they collected dust from 155 different rooms and did microbial diversity studies of them, and then tried to compare and contrast them. And again, before sequencing got cheap a couple of years ago, I think the most I'd ever seen anybody do with culture-independent studies of a building was 2 samples, pretty much. Like one indoor part and one outdoor part, or one. Literally, the scale at which people are doing this has run so fast that we have no intermediary sort of studies. There's, someone, Norm Pace, the guy who invented culture-independent studies, did a study of shower heads, the microbes in shower heads. And that's it. They didn't compare it to any other place. They did the shower head, and it's really interesting, but they didn't say, how does the shower head differ from a sink head, differ from the bathtub, differ from And that's because when they started that showerhead study 7 years ago, it was, $100,000 to sample one showerhead. And now it's $5 to sample one room. And so it just changes. So, the shop vac is actually expensive compared to the microbial sequencing that's being done. And I don't know anything about these classification systems, if this differs from what other people do. They say they've used it for other Oregon, it's the University of Oregon space type codes and definitions. There's probably some 200-page document that defines all of these things. And then this figure, which I still don't totally understand. So they also tried to classify spaces by this network analysis. And I guess this is a common, this is a common thing in building design analysis to try and understand the centrality of a room and the, they call it betweenness of the room, how much travel basically it is. Is it going to be between places that a lot of people go, or is it going to be off in a corner, basically? This is the way they basically calculate this, but they end up with which is most useful for their purposes, some numerical value for each room on this betweenness scale.
Was any of the motivation for the Sloan Foundation to do something, make some gesture away from the robustness of multiple disease-resistant pathogens in hospitals and operating rooms?
Well, so no. So here's how the Sloan Foundation program started, because I was there when it started. They were involved in biodefense projects. They got interested in microbial detection related to biological weapons attacks. And one of the reasons they got interested in that is they were funding, and still are funding, a large program in what's called synthetic biology. So in the last 10 years or so, people who want to engineer an organism can now do that in a way that some people consider sort of fundamentally different than the old school genetic engineering, which used to be used to transfer a single gene into an organism through many months of work and complicated steps. Now you can synthesize a piece of DNA in the laboratory and insert that into an organism. And they've been funded, a lot of people think this has enormous potential for all sorts of research areas and applied areas. The Sloan Foundation funded a lot of work on the security issues associated with this. They were worried that, they weren't really worried that someone would do something bad, but they wanted to make sure that people talked about the potential for someone to do something like synthesize the 1918 flu or synthesize something nasty. And so they funded a bunch of workshops, which I went to many of these, on, how could we detect if someone is doing something bad with synthetic biology? And the main way that people talked about detecting it was, looking at DNA data. And then people said, well, let's suppose there's a biological attack with an organism made by synthetic biology. Could we detect it? Could you, put out your shop vac and vacuum the air in the New York City subway system and figure out that there was a biological attack? So they funded a project to look at air in buildings. One of the first studies of using sequencing technology to look at the air in buildings as a way to see if you could diagnose if there was, some type of attack going on. And immediately everybody said, well, how could we do that? We don't know what's supposed to be there. We don't know what the background is. So if you want to detect an anomaly and you don't know what the background is, it's impossible. And so the Sloan Foundation started funding a few people to start to look into the microbial ecology of buildings, largely because they wanted to know what to compare to in case there was something going wrong. And that's how they got into this whole area. They realized when they funded the first 2 or 3 of these projects, including Norm Pace's Showerhead project and Craig Venter to sequence the air in a building in New York, that this was a whole field that was a gap. And the Sloan Foundation loves these gaps between fields where nobody is working. And they decided, after a couple of years of meetings, they organized a whole program in this, and it became really the microbial ecology of buildings. Now, if you ask the people involved, what is the justification for this? Biological defense is no longer one of the big justifications. The big justifications are things like you just said at the long answer to your question: hospital-acquired infections, mold in buildings, pathogens in cruise ships, food production facilities, and so on, and whether or not you could use the ecology of the community to help limit the probability of those things happening.
Is that just their disinformative front, or are they actually?
That's what they care about. No, they're not that is what they care about. The biodefense stuff is no longer there. I mean, Sloan Foundation wants to fund things that no one else is working on. DARPA is putting $2 billion into biodefense. They don't have to, there's nothing Sloan can do compared to that. So there's no point in a foundation that could put $20 million into something, funding something that there's already $2.5 billion in. It just doesn't compute. The Homeland Security put $1 billion into detecting. They had a $1 million prize for methods to detect biological weapons attacks from DNA sequence metagenomic data. They had, so Sloan moved into just the ecology. And that's what they funded. All of their projects relate something to ecology with, the hope that they will be useful for some applied purpose. So NICUs, the second study, is like the #1 thing that everybody is talking about. Can we understand the ecology of a hospital to try and protect babies who are born premature from getting these horrible infections that many of them get?
You walk away from this thinking that the hospitals are great incubators of disease-resistant microorganisms.
They are great incubators. We know that. So that part of these studies is, we know that. They're bad places to be mostly. So here's the description of the catalog number that they ordered a shop vac from shopvac.com. I love this. And then that they fitted it with a special, additional filter. They collected the samples over just a couple of day period. So that's another reason to use, something that has a high throughput vacuuming system. Although they admit that this is where they say, since the building was completed in 2003, that some of the dust may be really old. If you're interested, we can talk about the workflow that they used. Everybody uses a different one. Theirs is not that different from many of the other ones out there, but sort of standard compared to what we talked about with the workflow. Ribosomal RNA, PCR, Illumina sequencing, identification of OTUs, phylogenetic analysis, feed it into Chime software package to do Unifrac, and then they added a few bells and whistles on to the end that some of their figures are about, but they basically use the same general protocol.
So I have one question.
Yeah.
In their workflow, they do mention that they use DNA quantification.
Mm-hmm.
And I wasn't sure why, or like what that does.
So, yeah, that's a good question. If you have many samples and you do ribosomal RNA, the gene for ribosomal RNA, which is called ribosomal DNA, PCR, you end up with these, small tubes with PCR products in them. And then what you're going to do, depending on the method that you use, where you do this varies, but you're going to pool these together into one sequencing run on the Illumina system. And imagine if you had 100,000 bits of ribosomal DNA in this sample, and 10 in that sample, and 1 billion in one of these samples. And then you take out, you know, 1/10 of each of these samples. If this has 1 billion ribosomal RNAs in it, when you pool it all together, all of your sequences are going to come from this sample. So what you want to do is normalize so that you pool together equal amounts of DNA after the PCR, so that you get roughly equal number of sequences from each sample. Well, you can control it. If you wanted 10 times as many sequences from that sample, you could pool and put in 10 times as much DNA. So really, and in addition, when you run the methods to do the actual Illumina library prep and sequencing, you kind of need to know how much DNA is there in order to optimize it. So it's not just for the pooling, but it's really important for this pooling to have roughly equal amounts of DNA.
Each sample is represented.
Yeah.
Yeah, I think they did that in this one too. They said they rarefied the sequences.
Yeah. Well, so they do both, which is important. So they pool together everything to try and, before they do the sequencing, to hope that you get equal number of sequences on the machine, and then afterwards you try and normalize. They call it rarify, so that you do all your analysis with, say, 10,000 sequences from each sample.
Okay.
So you do both, try and make sure that you get roughly equal numbers, and then you still normalize across the samples. Does that basically make sense? So, and that's, there are places that try to skip those steps by making everything as uniform as possible. So if you use the exact same amount of starting DNA and run the PCRs in exactly the same way, you can try and skip these steps. It's generally a bad idea. And then everybody has a different visualization system, of course. Every single place, you know, has a different general way of doing this. And what this basically shows is something similar to what's in the other paper, which is they show one figure for the taxonomic assignments for the samples, and they show it for different taxonomic resolutions. So the top part of this is at the phylum level, and then they do it at the class level, and then at the order level. So they took their sequences that came out, and they assigned them to some taxonomic, domain to kings play chess on fine grained sand, to some taxonomic hierarchy, and they're going to show you the results for the phylum level, for the class level, and for the order level. In the Banfield paper, they show phylum and family, and then a little bit, I think, on species level classifications. So, you know, everybody, depending on the system that you're looking at, there are different areas of focus. But that's all in the data that you would get back out at the end on the taxonomic classification and the total numbers. So they're counting up for, you know, each of these samples. So this over here is building support. Can't quite read on circulation. You can probably read it better up there. And anyway, offices over here, office support, restrooms, etc. And now the relative abundance of ribosomal RNA reads that were assigned to these different taxonomic groups. Now this is all visual here, right? There's no diversity calculations. This is all just to show you the big picture of the results and let you make your own inferences based upon this. One of the things that they point out that they were really interested in is that many of the sequences were assigned to this Deinococcus group in some of the samples. We can talk about that if you want, but you know, there are some things that they got out of this taxonomic list approach that they did not get out of doing alpha diversity or beta diversity or big, you know, sort of mathematical calculations. And that is, for example, Deinococcus, the organisms in this group are extremely resistant to desiccation. So it's not that surprising that they start to show up in certain rooms when you're circulating the air inside for a long period of time. If you don't have a lot of water in there, maybe you're starting to select for organisms that are desiccation resistant.
There's no representation for Staphylococcus aureus. Is it a subspecies?
It's in the, Staphylococcus is in the Firmicutes
Oh,
phylum.
okay.
So Staphylococcus is a genus level classification, so it would be in the Bacilliales, I think. Yeah, so I mean, if you know something about a particular group of organisms at the genus level, then phylum level classifications don't always help you out. And again, this is just showing you some statistical analysis of all of those different variables that they measured. So they did not do the standard PCA for this part that we talked about. They do this Permanova test. I couldn't tell you exactly what it is, but it's described in the methods, but it's basically the same general idea. They're doing comparisons of the diversity between samples. They're asking how does that diversity correlate with these particular variables. They measure the correlation coefficient, and then they calculate a statistical probability that that is better than random. And that's what's shown in this figure. And so really, this whole list, these things apparently are all better than random, but some of these correlation coefficients are pretty bad. There's not much of a relationship between some of these variables. The only two they talk about are these two, space type and the air source air handling unit as strong signal of affecting the variance between samples in the microbial community. Again, all these other things have some detectable influence, but it's very subtle, very small. And then they do a bunch of other types of analyses to try and tease apart. And the band for the second paper, the Banfield paper has another approach to doing this. Basically, this is trying to tease apart now the relationship between sites and organisms that may dominate those sites and how those relate to each other. And so I'm not sure exactly the math behind doing this, but you can basically imagine that you're taking the sites, the samples that you have, you're looking at the relationship that they have to particular environmental parameters, and then you're asking if any of those relationships are dominated by particular taxa present in those samples. So if you have two sites, and let's just say one is office space and one is bathroom, and they're really different, you can then ask for the data embedded under that if that difference is due to any particular taxa difference between the samples, like the Staphylococcus between the samples or the Lactococcus. And that's what they're basically trying to show in this figure.
You mean if the difference exists because of due more to like one particular group than to
Yeah, so there are multiple ways to do this type of analysis. So you can ask basically, are there I didn't look at how they did this exact analysis, but you can basically ask if you separate out samples into one of these multiple dimensional spaces. You can ask for the samples that cluster in this part of the space. Are there any taxa that also show that pattern? So right when you do,
okay.
does that make sense?
Yeah.
So when you do all these analyses and you calculate, you have now many samples and you calculate distances between samples, and let's just say, some of the samples all cluster together.
Right.
You've thrown away, when you calculate that distance, you actually ignored the raw data behind it. But I mentioned before that you can sort of back calculate there are multiple ways to do this. You can basically try and figure out what parts of the data are leading to this pattern. So it could be that this is all due to differences in the presence. These could be present for staff, and these could be low staff levels. And that would lead to them to be separated out in this distance space, basically. I don't know if people read it in more detail. They might know more about what they did exactly here. But the reason I sort of skipped over it is in the literature I've seen like 50 different ways that people are doing this type of analysis, and I can't quite keep track of them, but I understand the general principle. And then they did this, what I found to be really interesting, and this may have been why they picked the building. So in this one building, they have 73 of the 150 or so sites that they sampled are offices. And they like this because rather than having 150 different types of rooms, they have 73 rooms that at least for some parameters, they're sort of, people are sitting a lot of the time. There's not a lot of traffic in those spaces. The airflow is consistent across them and so on. The doors don't open and shut a lot, et cetera. So now they want to use these to tease apart in a finer scale the causes of variation between sites. So when you're comparing bathrooms to offices, there are going to be 200,000 things that are different between them. And yes, they will cluster in different parts of one of these cluster diagrams. That doesn't really tell you anything because they have so many different environmental parameters that of course they're going to be different in their microbial community. And so does that make sense what they want to do with offices? It's very similar to what someone would do with sort of a controlled experimental study where they take C. elegans in the laboratory and they give them slightly different food but keep everything else constant and look at the effects of food on the microbial community. So they're using the offices in that same way. They do a million different analyses. Some are shown here. I think some are in supplemental information. To try and tease apart what's going on to cause the differences between offices. And basically their main conclusion is that the number one, and this goes back to the number one variable here, is sort of ventilation and air handling is causing the biggest differences between the office spaces. I was just going to try and figure out if this was actual, I don't know if people looked in detail, was this actual principal component analysis or was this the, I don't know. But again, they're separating samples out by just some metric of the microbial communities, and then they're overlaying, in this case, ventilation information, window versus mechanical, onto the samples, and there's a clear separation of the two.
NMDS, which is similar but different.
Yeah. Steve Kemble, the guy who was the first author in this paper, writes code with this software package called R. And when he was a graduate student, he designed a lot of software packages for doing comparative analysis of communities of plants and animals. And he has, like, in his head, as well as access in this code base to every possible way of comparing communities. So in papers that are written by him, there's usually like 10 new methods that you've never heard of that are probably better than any of the methods you've ever used.
It is better.
Yeah, right. So I mean, I'm not surprised. He's incredibly, incredibly smart and really good at using R. And he makes all the code openly available and posts it to this Picanti website that he's part of some software development consortium. And then they focus in again on a couple of taxa. I think these were the two taxa that had the biggest differences between the samples. One is Deinococcus, the one I already mentioned, and the other is this Methylobacterium. They do try to come up with explanations for the two of them, but they haven't, it's just going and looking up what these organisms might be like and trying to come up with a hypothesis for why they're different between these different communities. The Deinococcus one I know about because I used to work on Deinococcus. I don't remember what they said about Methylobacterium. Anybody remember what they said for why they proposed Methylobacterium was different between the samples? I don't remember. Oh, yeah, okay, so it's a plant associated, yeah. So if the air is coming from the outside, they're getting plant. And then even though this shows just a very weak pattern, I love this. So standard classic ecological studies have for years looked at this thing called distance decay relationships. So if you compare communities on islands from each other, and you ask, how similar are the communities to each other? And you plot how similar are they versus how far apart the islands are. There's what they call a distance decay relationship. The further physically they are apart, the less similar the communities are. And this is basically an evidence for certain types of biogeography, biogeographical history between the communities, and you can do this with latitudinal gradients, and you can do this with lots of different organisms. About 10 years ago, people were first starting to show this for bacterial diversity. Before that, they had shown that you see these patterns for other, like fungal diversity, and they even see it within one of these buildings, basically. So the distance that they use is basically the number of doors that you have to open to go between rooms. So they have this map. They can calculate for every pair of samples how many doors you have to go through to get between those two rooms. And the more that you have to go through, the less similar the communities are. So there's gradients basically in their communities, and the gradients are in part related to how much mixing there is between samples.
Is it actual just raw distance, or is it?
They actually
Culturing that happens on doorknobs.
No, they're asking how many. Oh, well, they're asking how many doors are there, as though that's the measure of how likely it is for them to mix. It's not. They're not looking at doorknob communities, right? They're looking at dust that collects.
People handling X number of doorknobs.
It's probably not that. It's
not that.
Very unlikely. It's probably that there's different microbes in different parts on average. There's one door that goes to the outside and it gets some plant associated microbes, and there's gradients, and those gradients are affect what's in one room you get from your source, and then the room next to that room is going to get some things from the same source by just mixing.
Have you ever noticed that doorways are anti-entropic, that they're where people aggregate, especially if you have to get through it yourself?
No.
That's why I see them.
But yeah, now I'm never going to look at them the same.
I suppose.
Yeah.
This paper summarized by saying that, in light of this evidence, that people, will enter an enlightened period of architecture. They didn't say that, but I mean.
No, but they quoted Churchill.
Churchill.
They did. They quoted Churchill that our buildings influence us.
Yeah, but did they do any kind of a census of sick days?
There's no. This is not about the health of the occupants.
But that was one of their overarching.
Yeah, but they don't have that information here. This is the preliminary. This is like when you go to an island for the first time. They're getting a layout of the land.
There's no Hilton there yet.
Yeah. Another thing which we don't need to go into is in many of these papers, they put the most complicated, confusing stuff in the supplemental information. So they have all these calculations that they did of different correlation coefficients for certain types of the data, but I don't want to go into in detail, so we can talk a little bit about the other paper. They also show these plots, which I really like. I like these more than so this is their way of summing up the taxonomic information across samples, but I don't like these bar charts particularly. There's this new approach that a lot of people are using called chronoplots, which show taxonomy at different hierarchical levels. So this is proteobacteria. I don't know if I can see it, and then Deinococcus over here. And it's just a really quick way to get a better idea for the different hierarchical level classification variation in a sample. So I think a little better than a bar chart. And that's dumped in the supplemental information. Well, colorblind, the bar charts don't necessarily help either, but yeah. And then here it is, the Churchill quote. We shape our buildings, and afterwards our buildings shape us. Anyway. And then the second paper is, in concept, the same idea. Looking at a building. Now they're looking at the occupants of the building too, and trying to relate what's in the occupants in this case to what's in the building and the different parts of the building, basically. And what makes it really interesting is that it's in a NICU. It's in a neonatal intensive care unit. And there are probably at least 10 studies of microbial sort of diversity associated with NICUs going on around the country, probably more. The main reason for these was originally this thing called necrotizing enterocolitis, which is a horrible inflammation disease that a lot of premature babies in particular get, and they can die in like a day from this necrotizing enterocolitis. They basically get a rapidly spreading infection in their gut, and then they're dead.
Did you say premature?
It's more common in premature infants. And so there's been a lot of interest from the NIH in particular in this necrotizing enterocolitis and what causes it and how to prevent it. There are studies at UCDavis now on treating premature babies with probiotics to try and prevent this being overrun by a pathogen. It seems to actually work. And prebiotics, which are food for probiotics. And that group that is doing this prebiotic-probiotic study has already published a paper where they went around a NICU and did some surveys of what was in the NICU. And there have been a couple of these out there, but this was the first study to really start to try and connect what's in the NICU to the infants in the NICU. Sorry, neonatal intensive care unit, sorry. And you know, a lot of different types of babies go into a neonatal intensive care unit. But in general, what they try to do in the NICUs in most of the world is to sterilize the environment. Because these babies are frequently premature or they have other health problems, they frequently don't have a fully developed immune system. They may have a congenital heart problem. They may have other things that lead them to be prone to infection. Many of them are born by C-section on top of everything else. And they pump them full of antibiotics frequently. And they try to make the rooms as sterile as possible. And they try and make the people coming into the rooms be as clean as possible. So, you know, many of the babies aren't breastfed because that, you know, requires touching them, God forbid. And so there's been now this model in the last few years that this may be a bad idea. Keeping the rooms completely sterile and certainly keeping the babies completely sterile and overrun with antibiotics may, you know, it does prevent some infections almost certainly, but it may set them up for other problems by not having a normal colonization. And so this study is sort of in that context, like what do these infants get colonized with in a NICU? And does it relate to what's in their mother or in the doctors, or what does it relate to? So they did two, I mean, it's not a big study, they did 2 infants, right? And they basically did the same type of thing. Again, we can talk about the details. They have a workflow for collecting samples, isolating DNA, doing ribosomal RNA PCR. They are trying to get full-length ribosomal RNA sequence products, so they actually amplify a larger region of the ribosomal RNA, and then they sequence them. But when you sequence So the ribosome RNA is about 1,600 base pairs long. You can PCR nearly the whole thing from a community. If you then feed it into Illumina sequencing, even the best Illumina sequencing will read something like 500 base pairs at most at a time. So you don't get the whole sequence, and you might get this randomly. So you would end up, if you fed this into Illumina sequencing and you had a pool, this 10,000 different ribosomal RNAs, and you get out sequence data, one of them would come from this part of sequence number 1, and one of them would come from this part of sequence number 2, and one of them would come from this part of sequence number 1, and one of them would come from this part of sequence number 7, and you get this non-overlapping or slightly overlapping data back of Illumina sequences from diverse organisms. And what they have is this software called Emerge that they use that tries to stitch these back together into full-length ribosomal RNA sequences that supposedly come from individual organisms in the environment. And they also do, which we haven't talked about a lot, and we don't need to go into a lot of detail, they also do metagenomics. So they randomly sequence the DNA from these communities, and one of the things they do is they run that same DNA through their ribosomal RNA assembly program, and they get full-length ribosomal RNAs that are not by PCR, that are now by random shotgun genome sequencing. And they do many other things with the metagenomic data, but mostly they focus on the ribosomal RNA data. They did do the sequencing at UCDavis. I don't know if people noticed that or not.
So how do you pull out the sequences
From
from just 16S?
the metagenomic data, you just do a similarity search. So like BLAST or some other similarity
So you
search.
sequence the whole thing and then pull out the...
Well, they used all the data for other parts of their study, but one component of their study, they're just scanning for the 16S ribosomal RNA genes in the random shotgun. So they collected a lot of samples associated with infant 1 and infant 2. They then ran it through, with their ribosomal RNA PCR. They then ran it through this Emerge tool, and they assembled back together either 13,000 or 11,000 different ribosomal RNAs from across these infant samples. And then they do a bunch of stuff with either this data or with the metagenome data. So one thing they focus on is different from the other studies. So the Oregon study is a snapshot in time, right? They go in with their shop vac, and they suck up some samples, and each room gets one sample, basically. And in this study, they went back and collected, in essence, the same samples over and over and over again from the rooms and from the infants. And now they have time series on top of different samples. They have samples over time. They asked some questions about the multiple samples that they collected. Alpha diversity metrics and the variability in the alpha diversity metric. I think there's a, yeah, so, and then they show you some of these samples over time. So they scale the time relative to when the infants were born, but they also collected, it's not just from the infants, but they collected samples from the rooms. Anyway, there are a bunch of different places in the facility, and also eventually they will look at the different sites within the infants. This is just the facility shown here. And if you zoom in, the first part of the figure is showing it divided by phylum. So here's the different phyla that are shown. Oh, now you can read it. Electronics, hands, incubators, sinks, surfaces, and tubes for infant number 1. Electronics, hands, incubators, sinks, etc. Some they didn't get any product from, so they swabbed, they did PCR, and it didn't work for some reason. They may, I don't know why for some of them, but there may just have been almost nothing there. An important thing for both of these studies, they are not showing actual abundance of organisms. They're showing relative abundance. This does not tell you how contaminated a NICU is. right? Even though many of the press reports on NICU studies always focus on finding bacteria, they're almost always not counting how many bacteria there are. They're comparing the relative abundance of proteobacteria versus something else. It could be that there's 1 bacteria per square meter. I mean, well, it's got to be more than that because they're not swabbing a whole square meter. But the total amount of bacteria could be still very low in these samples. But anyway, you can see, for example, the sinks, almost everything is proteobacteria in sink number 1 and sink number 2. And if you can sort of compare them, here's surfaces. The surfaces are reasonably diverse. Hands, they didn't get a lot of data over here, but incubators, you can see sort of the same, some of the same phyla showing up. So at the phylum level, you don't see that much differentiation. You see a little bit more when you zoom in. Now this is the family level. So here's the family for the sinks for infant 1 versus infant 2, and these are different time points. So, there's lots of scatter. It changes a lot over time. That's important to realize. But, on average, you see the same families showing up over the different time points in these sinks, and the same families here. And then in surfaces, there are different families than in sinks, but the different surfaces over time have the same families, different relative abundance of those families. So this is giving you a picture of the variance within a type of environment over time and the variance between environments. And then this is, they're doing actual standard principal coordinate analysis, I think with CHIME, just like the whole workflow that we talked about. And now they're clustering samples for infant 1 and infant 2 and looking at how they fit relative to the metadata. So, this is, when you show this, it's fine. And again, it's useful if you know something about particular taxa or you're interested in something about the taxa. But to me, this is what you should start with in most studies, is the overall similarity of communities to each other to see if there's any general trends. And there are some general trends here, but there's certainly some, like here in infant 2, these samples, which are tubes, there's nothing in between them. And here's the fecal samples. But here, fecal and tubes sort of mix together a little bit for infant number 1. Here's sinks. Sinks stay somewhat isolated relative to other things, but here's a big cluster, electronics and hands and incubator and other things. So there's not complete separation between these different sample types.
What does it mean when sinks are more or less isolated in both infants, but it's in a much different spot in infant 1 or infant 2? What is that?
I think it means nothing.
Okay.
It's a mathematical way of converting this multidimensional space into a 2-dimensional pattern. And I think when they ran this, it's a different run. If they had shown it on the same plot, they could have pooled all the data onto one plot. And then if they were in different places, that would mean that sink from infant number 1 was really different than sink from infant number 2. But I don't think you can compare from, does that make sense? Yeah. This is completely cool, and it will take me like 4 hours to try and explain it, but I will basically explain it as what they're trying to do is look at now the genome data from these different organisms. So they're getting random shotgun genome data. They map genomes to particular taxonomic groups. That's what the colors correspond to. And they also run this complicated thing called a self-organizing map, which takes all the individual assemblies that you get from the genome data and puts them into this 2-dimensional space, tries to group them together by pieces that are similar to each other in their nucleotide composition. So it might be, like GC content, and puts them into a similar place. And they basically use this to try and sort the data from a community into which taxa it likely comes from. And that has nothing, this is just a cool picture for this paper. It has nothing to do with the conclusion here. The conclusion here is basically you should just look at the taxa. And the taxa list here that they're able to sort a lot of the genomic data into is really different from the taxa list here that they're able to sort the genome data into. So infant number 1 and infant number 2 have really different species. When you're able to assign individual reads to like some species level classification, really different organisms inside the guts of these two infants. This is just more data on the genomes. This is the equivalent figure to that other one with a different method for now, again, trying to relate samples to taxa within those samples and the samples to each other. This is the total data, this massive hairball. They then zoom in on just a subset of the data. I'm not sure if it's that useful to go into. Again, this is one of the ways that people are now trying to go beyond just clustering samples to retrieving information about the taxa that might be responsible for some of the clustering of samples. And then this is the part that I find completely kind of mind-blowing and interesting, which is comparing infant 1 to infant 2 now. This is showing pretty clearly the different taxa that are in infant 1 and infant 2. It doesn't matter what these individual ones are, but you can just see by the color scheme that it's really different organisms here than here. So really different taxa colonizing infant 1 and infant 2. And then this is the cool thing. Here's the environment for infant 1. These are those same taxa, the relative abundance of those same taxa in these different environments. And there's just, what they conclude from this is that it's likely that infant 2 is getting colonized by microbes in its environment. And infant 1 is getting colonized by microbes in its environment, and they're different from each other. Which, yeah.
How do they know that it's not the other way around?
They don't.
Okay.
They have no idea. They're not, yeah, I mean, if they had identical twins, so interestingly, these are both born by vaginal birth. Both of these infants should have been colonized somewhat by birth, and yet there's still this relationship to the environment. So it easily could be the other way around. I mean, the babies are getting touched, the doctors are then touching the environment. I don't think there's any directionality that they can conclude from that. That's my impression.
One of them was premature or critically underweight.
Yes, but born by normal vaginal birth, not C-section.
I mean, they would be in. I mean, this is just straight off the side, but they'd be in different parts of the hospital.
They were not in the same. I think there was same NICU at different times, so they were not there at the. I think they were not together.
So is there some speculation that the immune system then in infant 1 is, quantified?
Their speculation is that it just relates to what's in their environment, but they don't. Yeah.
One of them had a twin, right? So did they try to compare
Healthy ones,
between both of the?
I didn't see any data from that. I don't know if they didn't. They probably didn't have approval for.
Infant 2, one of the first tables said it was from twins.
Yeah, but I don't think they have any data. So I think they had IRB approval to do a study of the babies in the NICU, and they weren't allowed to ask even for getting data from a baby not in the NICU.
That would have been a great control though.
Yeah, sure, but this is the challenge of doing human studies is getting informed consent and getting approval to do anything doesn't always give you the flexibility when one of those things comes up. But we can add, I mean, I know Jill Banfield, we could ask her if they ever got permission. It'd be really interesting. So then, this is just sort of their attempt to, again, show you a visual representation of the connection between infant 2 and its environment and infant 1 and its environment. You can trust it or not trust it. what they then try and do is run a more statistical analysis of this type of data. And what they do is run this program called Source Tracker. And what Source Tracker basically tries to do is connect samples to environment. It can connect, if you have just some arbitrary environment with lots of samples.
Yeah, they use the same thing in the paper I read.
And you have some classification scheme for those. Let's just say this is office and bathroom and house. You can basically ask, if you take one of these samples, or even one of these from those classified samples, and you can ask for its community, what proportion of the community, can we build a model that says that, some percentage of the community came from houses, some percentage of the community came from bathrooms, and some percentage of the community came from offices? And what are those percentages? And it's, this is a common thing that's been used in studying epidemiology of infectious diseases for ages, people trying to figure out the source of a particular contaminant. And what they're doing with this method is looking at the sources for communities as opposed to individual organisms. And I don't know, maybe did you figure out how Source Tracker works? Yeah. I don't remember, I used to know it. I don't remember exactly how they do this. But that's what they're trying to do here is they say that where does infant 1 at this time point, where does it likely get its community from? And, X percentage came from tubes, a lower percentage came from surfaces, and this amount came from electronics here. The closest match was to tubes. And then some of the samples, there's unknown. So Source Tracker allows you to ask, so here's a sample. If it's really unique, some percentage of the sample, it'll just posit came from somewhere else that you don't have data for. And so as you get later into the life of this infant, more and more of the sample on average comes from unknown sites. And then here's infant number 2. For infant number 2, a lot of it is coming from the tubes. In infant number 2's environment, doesn't have the same pattern with more of it coming from unknown places over time. But this is basically another way of looking at this type of data to try and say that there's some connection, let's just say, between those types of samples. And again, it doesn't really tell you, it's called Source Tracker, but there are ways you could have this be source receiver too, depending on how you set up the analysis. But anyway, I find this figure showing sort of the taxonomic distribution I find to be really interesting, that at the fine scale resolution, infant 1 and infant 2 are different from each other and more similar to their local environments. And then they do this last thing, which is whole genome phylogeny. And the reason they do this is when they do this type of analysis, almost all the taxa are different between infant number 1 and infant number 2, except Enterococcus fecalis is shared between infant number 1 and infant number 2. So they then say, okay, we have some metagenome data, genome data. Can we tease apart Enterococcus fecalis in infant number 1 versus infant number 2? Are they the same as each other? Or can we get an even finer scale resolution, basically, than we were able to get with the ribosomal RNA data to say, something about these two Enterococcus strains? And when they do this, they see that infant in bold are infant number 1 and infant number 2, the Enterococcus fecalis from them. with two different sort of approaches, they're basically identical to each other. I mean, they talk about how they are able to sort of tease them apart a tiny bit, depending on which region of the genomes they look at, but they're very, very, very closely related to each other, as opposed to the rest of the taxa, which are, just completely different genera even showing up in the different organisms. And then the last thing they do is, there's no figure for it, but somewhat speculative, but pretty wild, which is they scan through the metagenome data. So they got random metagenomic information from these organisms. And they search for genes that may be involved in resistance to antibiotics, basically. Because that now gets to, this is their connection now to hospital-acquired infections and antibiotic resistance. This one little paragraph at the end of the paper. I wouldn't make much of this. They didn't do, comparisons across, infants that were not in a NICU. They didn't do, comparisons to the twin, or they didn't do, so the fact that they found numerous antibiotic resistance genes, as well as some things that might be involved in resistance to antimicrobial compounds in the NICU, no idea what this, I have no idea what this means.
Wouldn't that put them on the line for making a determination if you, under current methods, if you continue to, what's good procedure to try to eradicate or control populations?
Well, the problem is, I think if you went to any microbial sample on the planet, you would find these genes. And I think if you went to healthy babies, not in the NICU, you would find these genes. And I think if you went to babies born by a midwife, you would find these genes.
So did you make any call on whether or not you should change the way that you try to control or eradicate?
They could say that, but they have no data that has any connection to that whatsoever. So I think this is basically showing that there is potential useful information in the metagenomes. But I don't think you can conclude anything from what they did with it. And just two infants, you can't really conclude a lot from study of two individual infants, especially when they didn't compare to healthy infants in some other facility or et cetera. This is, again, the building study is probably more comprehensive because they had more samples and they had more ability to tease apart the variance between samples. This is sort of the true, first let's gather some data on what's in infants over time in a NICU. And two samples is nowhere near enough to make any big scale conclusions about. I mean, it's interesting that this to me is the most interesting part. Again, two samples, maybe if they did 20, you wouldn't see this, but to me, that the infants that they looked at, their gut communities were most similar to things in their actual environment is very suggestive of mechanisms of exchange between infant and the environment. And does suggest, I think that, NICUs, first of all, aren't anywhere near as sterile as people thought. We don't know the total numbers of organisms, but they can swab the organisms, and those organisms are not random. They're similar to the organisms in the infant. So either they're colonizing the infant or they're getting out of the infant, and the NICU is not clean. So anyway, wait and see when they have 100 babies.